Good evening. Now we are starting with the next class of LPT. Today we will study about the cleaning and sanitization of dairy equipments and plants. Okay, it is a very important topic in dairy technology and only 2-3 questions are asked, but those questions are important because sometimes we tend to forget those things. So today we will study about basic definitions of cleaning, sanitization, sterilization, soil, millstones. Then we'll proceed towards importance of cleaning and sanitization. We'll classify about the different detergents that are there and study about their properties. We describe the sanitizers and their desirable properties. Then we'll study about the cleaning in place systems and various steps. Then solve some of the MCQs. Okay. Then starting with the key definitions. So what do you mean by cleaning? Cleaning is removal of the dirt, visible soil, residues, etc. using physical and chemical means. And what do you mean by sanitization? So it is reduction of the microbial load to a safer levels, okay, which does not mean sterility. Sterility means majority of the microbes, all of the microbes including spores should be completely destroyed. Okay, whereas sanitization means reduction in the microbial load. Okay, then next is the disinfection. So when we consider about the disinfection, we are mostly focusing on the inanimate objects, okay, non-living objects. Okay, so it is destruction of the most of the pathogenic microbes but not their spores, because spores are still present in case of disinfection. So these are the basic definitions of all these terms. Okay, next coming to the soil and milkstone. Okay, so what do you mean by dairy soil and dairy milkstones? Okay, so any unwanted material which are present on the dairy equipment surface, which include fat, protein, lactose, mineral, microorganisms. Okay, so these are combined together and present on the dairy equipments. So we do not want that, because we want our equipments to be clean, to be sterile, okay, to be sanitized, and so we want to remove all these objects from the dairy equipments. Okay, once we have used our dairy equipments, daily we need to clean it. Okay, we need to sanitize and sterile it. So for that we need to remove the dairy soil. So different types of soil can be there: fresh, which is easy to clean, and aged or dried, which becomes harder and it is very hard. Okay, it is very difficult to clean such type of dairy soil, and some are burnt on soil, so it is hardest and the most difficult form of soil to clean. Okay, so it is very difficult, and because it leaves protein residues from heat exchangers. Okay, it will leave its residue and it will not be removed by usual cleansers. Okay, then what do you mean by milkstones? So milkstones are hard yellowish white deposits which are present over the equipments. These are composed of calcium phosphate, calcium carbonate and denatured protein or denatured fat. Whereas in the case of dairy soil, it was fat, protein, lactose, minerals and different kind of microorganisms. In the case of milkstones, these are calcium phosphate, calcium carbonate compounds. Okay, denatured protein, denatured fat compounds. Okay, these are formed because of repeated heating of milk and inadequate cleaning, because we are not cleaning it properly, and repeatedly heating of milk, due to which the calcium phosphate, calcium carbonate is being denatured along with protein and it is depositing on the equipments. Okay, because cleaning is not being done and repeatedly we are heating the properties, heating the milk and heating the equipments, because of which the calcium phosphate, calcium carbonate, these will deposit and along with the denatured protein and fat, and then we call it milkstones. Okay, so how we'll remove it? Using acid cleansers. Okay, acid cleansers will dissolve all these calcium phosphate and calcium carbonate. So what are the examples of such acid cleansers? Nitric acid, phosphoric acid, HCl. Okay, all these acid cleansers, what they'll do? They'll dissolve the calcium phosphate, calcium carbonate alongside the denatured protein and fat, and then it will remove the milkstones. Okay, so milkstones are acid soluble deposits. Okay, these are acid soluble deposits. You must be knowing about hard water. ठीक है, hard water or the water stones which include calcium carbonate from hard water, but these are not milk stones, because in case of milk stones denatured protein and denatured fat will also be there. Okay, and both are removed by acid cleansers, because acid cleansers will dissolve the carbonates and phosphates, and milk stone are called the milk scale or the boiler scale. These are another names. Then next, coming to the importance of cleaning and sanitization. First we studied about the definitions, the basic properties, why they are there, ठीक है, why milk stones are formed, from what compounds they are made of, and how we will remove them. Now we are studying about the cleaning and sanitization, the significance of cleaning and sanitization. So first we need to improve the quality and safety of our equipments, ठीक है, to reduce and to prevent the microbial contamination, to maintain specific plate count, okay, coliform count within limit, okay, within legal limits. We studied about these limits yesterday, and today we are studying how we'll ensure that there is no microbial contamination after using of these equipments. Then to also improve the efficiency, because if there will be milk stones, if there will be milk soil, then what will happen? It will reduce the efficiency. Okay, because how? Because it will lead to heat transfer in during pasteurization. It will lead to heat transfer. Okay, and so heat will be lost and higher energy will be required, and it will lead to lower efficiency of our dairy plant. Then next, it will also improve the shelf life of these equipments, okay, because after pasteurization also it will also improve the shelf life of our milk. It will also improve the shelf life of our milk, because it reduces the post pasteurization contamination and improves the shelf life of this pasteurized milk. Okay, then it will also lead to mastitis and contamination. Okay, milky equipments are very important, because otherwise if there will be inadequate cleaning, it will lead to teat canal infection and biofilm formation. What is biofilm formation? Biofilm has microbes. Okay, it will lead to contamination. Then next, we also need to improve the legal compliance. How we'll do that? We will ensure that there is no microbial contamination, SPC should be maintained, ठीक है, coliform count should be within the legal limits, ठीक है. So PFA Act, as per PFA Act, FSSAI regulations, so we need to have hygienic equipments, otherwise we will there will be cancellation of the dairy plant ka license, ठीक है. Then lastly, we need to improve the economic performance. So for economic value also, we need to improve and clean and use clean products. Okay, it reduces the product recalls, spoilage losses will be there, chemical treatment costs downstreams. So all these are the economic losses, and how to prevent all these losses? Through cleaning and sterilization, okay, through using sanitary equipments. Then next, what are the types of equipments? First we'll study about the detergents, then we'll study about the sanitizers. Okay, so what are the detergents? Detergents are the cleaning agents that reduce the surface tension and remove the milk soil from the equipment surfaces. Okay, so basic property of these detergents is that they will reduce the surface tension. So classifying these, first is the alkaline detergents. Okay, what are alkaline detergents? Sodium hydroxide, potassium hydroxide, sodium carbonate, all these are used to remove fat and protein, and these are the most common used. Okay, alkaline detergents are the most commonly used detergents in the dairy equipment plants. Second is the acidic detergents. So like we studied earlier, acidic detergents dissolve the milk stones, so they are important for removal of the milk stones in the dairy plant. Examples include nitric acid, phosphoric acid, HCl, okay, citric acid, gluconic acid. All these will remove the milk stones, the mineral deposits, water scale, boiler scale, etc. All these will be removed by using these acid detergents. Apart from the acid and alkaline detergents, we also have the neutral detergents. These are non-ionic surfactants and mild soaps. So these are just generally used for mild cleaning. So these are also safe for the workers, and these are less effective if they are only used alone. If we are only following the neutral detergents, then these are not much effective for the cleaning and sterilization of the milk equipment plants, but they are safe for the handlers, they are safe for the ones who are using them, they are safe for the workers as well. Then lastly, enzyme detergents, for example the proteases, lipases, which are biotechnologically used. Okay, so these are important for low temperature cleaning, membrane systems, because they are also gentle on surfaces. Okay, so enzyme detergents, neutral detergents, acid and alkaline detergents, these are the types of detergents. And basically, what is a detergent? It reduces the surface tension and it is employed for removal of the soil from the equipment surfaces. Next, what are the important properties, specific properties which make the detergents useful? So first is the good wetting property. So since we want to reduce the surface tension, it should have a good wetting property. Okay, it should allow the detergents to spread and contact the soil. Next is, it should have a emulsifying power. It should break down the fat into small droplets for suspension and removal, so that the surface area is also enhanced. Then first was the wetting property, then second was the emulsification action, then third is the deflocculating action. It should disperse the protein aggregates and prevent redeposition. Then the fourth property is saponification. So it should also convert the fat into water soluble soaps. Okay, alkali specifically should be able to convert it into water soluble soap. So it should have wetting property, emulsification, deflocculation as well as saponification powers. Alkali has good saponification power. The next, it should also be able to chelate minerals. It should also be able to dissolve these minerals. So calcium, magnesium ions, all these should be chelated. So how it will do that? It will prevent the water hardness. Okay, it will reduce the water hardness. Then next, it should also be able to rinse. It should rinse off completely. It should not leave a residue. It should not have adverse effects on the equipment. It should not be toxic or corrosive. It should not corrode the stainless steel or harm the workers at use concentration. It should not be harmful. It should not be toxic. And lastly, very important, it should be economic and stable for use. It should maintain the potency. It should be cost effective for large scale dairy uses. So these all 8 are the properties, desirable properties that we are looking for in a detergent. When we classify the detergent, when we use them in the equipment and the milking plants, then these should follow these 8 criteria. Then only we can ensure that we are going for better cleaning and sanitization. Next, coming to the sanitizers and the types and classifications of sanitizers. So what is a sanitizer? First we studied about the detergent. Now we are studying about the sanitizer. So what is the basic definition of sanitizer? So it is a chemical agent that reduces the microbial count to a safe level. Since we studied about cleaning, we studied about sanitization. We studied how sanitization means reduction of the pathogens and the microbial load. So sanitizer is an agent which reduces the microbial contamination to safe levels in the cleaning surfaces. So basically it should reduce the microbial load by about 99.99%. It should be able to remove the microbes up to 99.99% accuracy. So these are all the types of sanitizers that are used in the routine basis. First is the heat. So basically we are using steam and hot water at the temperature of 77 degrees Celsius for 5 minutes, or 100 degrees Celsius steam we are using. So what will happen? It will not leave any residue. These are the advantages, because it does not leave any residue. It is reliable, it is very cheap, economical, cost effective method. But what it does, sometimes it leads to corrosion, because it will corrode our equipments. It will also lead to energy cost, because we need to use the steam, direct steam we need to use. So sometimes energy cost will be lost. Then second is the chlorine compounds. These are cheaper and fast, broad spectrum. It will act on gram positive, gram negative bacteria. All kinds of bacteria will be affected, and it will reduce the microbial counts. And we are using it at a concentration of 50 to 200 ppm to ensure that the chlorine is available. Then what is its disadvantage? Because it gets inactivated by organic compounds and hard water. So we need to use some caution. We cannot use it in case of hard water and organic compounds. For them, what we can do is we can go for steam. We can use combination of methods. Then next is iodophores. Iodophores means iodine and surfactants together, called as iodophores. So the concentration is 12.5 to 25 ppm iodine should be liberated in the iodophores. So advantages include less irritant and it is indicator. So we can use color change. We can ensure that the color change is there. So it can show that the efficiency of equipment is there. We can show that it is efficient. But there is also disadvantage because it is very expensive and leads to corrosive action above 50 degrees Celsius. If the temperature will be improved, if the temperature will be increased, it will lead to corrosion. So it also has harmful effect. So apart from these three, we also have quaternary ammonium compounds, QAC, or quaternary ammonium compounds up to the level of 200 ppm. So issues include inactive against gram negative and it also forms film. But there are also advantages. Very routinely quaternary ammonium compounds are used in the field level, and because these are non-toxic, non-corrosive and odorless action is there. Apart from this, acid anionic sanitizers are also there, pH having less than 3. Why pH is less than 3? Because these are acidic in nature. So what will happen? It will remove the mineral film. It will be good for stainless steel. It will not react, not corroded. But sometimes some other metals, if they are used in the equipments, these will get corroded, because we know acid are highly reactive in nature. They will react with metals and these will lead to corrosion. But it does not corrode the stainless steel, and most of our equipments will be made up of stainless steel. So usually these are very important, very highly useful. Lastly, we also use peroxyacetic acid at the concentrate level of 100 to 150 ppm. So what are its advantages? These include effective at low temperature, no residue and eco-friendly. But the issue is again corrosive at high concentration and very expensive also. So these 6 were the classification. Usually what we follow, we use heat, we use chlorine, and we use quaternary ammonium compounds and also iodophores to some extent. Then since we also studied about the 8 properties of detergents, now we'll also study about the 8 properties of these sanitizers. Okay, starting with the broad spectrum activity. Since we want to reduce the microbial count, okay, so microbial count should be reduced, but these should have broad spectrum activity. They should also include bacteria, yeast, mold, virus. These should be anti-infective in nature. Okay, these should also reduce the count of bacteria, yeast, molds and viruses, which means these should have broad spectrum activity. Second, they should have rapid action. They should take very less amount of time to act on the microbes. So should kill microbes quickly at recommended concentration. So these should be rapidly acting. Third, these should be non-toxic, or they should not leave any residue in the equipment, should not have any adverse effect on the animals or the equipments. It should be safe to use levels, no harmful residues, and should not harm the workers or the handlers. Then it should have non-corrosive, non-irritant action. Should not damage the stainless steel, rubber gaskets and the plastic parts that are used in the dairy equipments. Then fifth point, they should be stable. They should have potency during storage. They should not get degraded by hard water or organics like we studied in case of chlorine. Then sixth, they should be economical. They should be cost effective for commercial dairy plants, and especially at the lower concentration also they should be effective and economical. Then seventh, they should be easily removable, easily rinsable. They should not form any foam activity. So cleaning in place systems use excess foam, interferes with flow. They should be easily rinsed off. Then lastly, they should also have indicator property, so that we know that they are acting, that their efficiency is there, that they are effective in the nature. Then visual indicator of activity like the iodophore will turn pale when they will exert their effect. So that is very desirable, so that we can know that the end point has reached and they are able to exert their effect. Then these are all the properties of the good detergents, good sanitizers. So firstly we studied about the definitions, then we studied about the cleanliness, and then we studied about its importance. We studied about the detergents, their qualities, types, and then we studied about the sanitizers, their desirable properties, and the different types and classification of sanitizers. Now we are proceeding further towards cleaning in place. This is very important when it comes to exam point of view, and usually questions are asked from this portion. cleaning in place. These are modern automated cleaning systems for dairy plants and it does not require dismantling. Okay, it can be used routinely without dismantling using circulated cleaning solution at controlled temperature, concentration and flow rate. Okay, so equipments are cleaned without dismantling them. They are cleaned at their own place and these are the following methods. Okay, this is the process through which the cleaning in place is being done. First of all, there will be pre-rinsing with water. It removes all the loose soil. Cold water is used for 2 to 5 minutes. Okay, then we'll start with the alkali wash. Sodium hydroxide 1 to 2% at 70 to 80 degrees Celsius for 10 to 20 minutes. It will remove the fat and the protein. Then we will go for immediate rinse, intermediate rinse, because we want to remove the leftover alkali also using warm water. Then again we will go for acid rinse using nitric acid 1 to 2%, then at 65 to 70 degrees Celsius temperature because it will remove milk stones. Then we will also go for again we will remove the remaining acidic water using neutral pH. Okay, using potable water until there is neutral pH, so that we will know that whole of the acid has been removed. Then lastly we will go for chlorine. We need to sanitize it also, so we will go for chlorine or quaternary ammonium compound because it has broad spectrum activity. Then flow velocity should be there, turbulent flow should be there, laminar flow should not be there because it does not show that it has been done effectively. So cleaning in place systems save water, time, chemicals, labor compared to manual cleaning. They are very effective and used in modern automated systems. Temperature, concentration, contact time and mechanical action, all of them should be very good. Okay, so we will ensure that the cleaning in place has been conducted effectively. So this is all for cleaning in place. Moving further for the principles. What are the basic principles that we are following for cleaning in place method? Okay, starting with the general cleaning sequence. So first we will go for dry cleaning. Remove loose soil without using water, sweeping. Then we will pre-rinse with warm water at 35 to 40 degrees Celsius. Then we will start with detergent solution. Okay, first alkali and then we will go for acidic detergent. Mechanical, then physical action which should also be there alongside. Then post rinse to remove the remaining detergent. Then we will go for cleanliness inspection. We will show that whether the detergent has been removed, whether it has been able to remove the physical agents, the soil, milk soil, the milk stone, all these should be removed. Then we will go for sanitization and remove the microbial load. And then final through chemical sanitizer. And then we will finally also go for rinsing and remove all these things. Okay, the temperature should be 60 to 80 degrees Celsius for alkali, 60 to 70 or 60 to 80 again for acidic. Then the concentration should be 1 to 2% for acidic for alkali and 0.5 to 1% in case of nitric acid. Contact time should be 10 to 20 minutes. For mechanical action should be turbulent flow. And water quality should be soft water because hard water reduces efficiency. It leads to formation of milk stones. Then soil type. Okay, the most important, fresh one, aged one, burnt ones, all of them should be removed. So for that we need to ensure that whole of them should be removed using cleaning in place system. Then we will also study about little bit about the milking equipments that we are using to have milk from the animal. So teat dipping is also important factor. Okay, the machine milking should be important, otherwise it will lead to mastitis and it will lead to infection of the mammary gland. So we need to reduce that, prevent that also. How we will do that? By ensuring that the milking machines and the teat dipping, teat sanitization is being carried out. For this we need to make the farmers also aware. Okay, so starting with the machine milking, immediate rinsing should be there to remove the milk residues before they dry, because we studied how aged milk stones, aged milk soils are most harmful and they are most difficult to remove. Once they get aged, it is very difficult because they get dried and they stick to the surface. Next, hot alkali wash should be there for the milking machines. Then again rinse with the acidic solution. Then final hot water rinsing. If we will fail, then biofilm formation will be there. Okay, the mastitis risk will increase. Okay, in the milk the bacterial count will increase, total bacterial count will increase because biofilm formation will be there and the mastitis risk will increase. So milking liner or cluster should be changed after every 2500 milkings. Then second is the teat dipping or the teat sanitization. Pre-dips and post-dips both should be employed. Before milking, teats should be sanitized because it reduces the environmental mastitis pathogen. For example, the E. coli, the coliforms, staph bacteria. All these are environmental mastitis pathogens which needs to be cleaned. Then post dipping also should be there after milking, most important because teat canal will remain open for 20 to 30 minutes post milking. So after and before both should be employed. Commonly used are iodine and chlorhexidine or hypochlorite solution. All three are very important and they are usually used. These are iodophores. Then barrier dip should be there, physical film, emollient, disinfectant should be used. Then intramammary infections will be reduced if we ensure that teat dipping and teat sanitization is being carried out, both pre-dip and post-dip. Then it should be checked for iodine residue because we should ensure that iodine should not come out alongside the milk. Then teat dipping with iodophore is the most effective single practice to reduce mastitis incidence in a herd. We are talking about the large commercial herds. So sanitizers, we studied about all the sanitizers. Let's have a quick recap of all these things, quick revision. So starting with the chlorine hypochlorite solution, 50 to 200 ppm is used. It is broad spectrum in nature, inactivated at more than 50 degrees Celsius temperature. So we need to ensure that it is used when we employ this, the temperature should be less than 50 degrees Celsius. So it is very fast acting, then cheapest. Then it also leads to organic matter inactivation. It gets inactivated in the presence of organic matter. This is the key limitation. Next, coming to the iodophores. So 12.5 to 25 ppm is being used. It is also broad spectrum in nature, but we should need to avoid the 50 degrees Celsius temperature. It is self-indicating color. It gets pale in color once it gets exhausted. It is expensive and sometimes also leads to stains because it is staining and it is coloring nature. Then quaternary ammonium compounds at 200 ppm pressure, at 200 ppm concentration. It is mostly active against gram-positive bacteria. It is very stable at different temperatures. It is non-toxic. It does not lead to rinsing. It gets easily rinsed off. Then it is not resistant. It is resistant against gram-negative bacteria. Gram-negative bacteria are not killed by the quaternary ammonium compounds. So sometimes biofilm formation will be there, and once the biofilm formation will be there, the total bacterial count will increase and the mastitis chances will further increase. Then peracetic acid solution, 100 to 150 ppm will be used. It can be employed for broad spectrum and even for spores it is very highly active. Then it is effective even at 4 degrees Celsius refrigeration temperatures. It is eco-friendly, does not leave any residue, but the issue is that it is also corrosive and sometimes costly in nature. Then again acidic and anionic, pH less than 3 can be used. Again broad spectrum, good and stable, but again corrosive. It removes the mineral film also. Then heat or steam, I talked about heat and steam, which is mostly used, very effective. All organisms are killed, energy cost is there, and sometimes corrosive action is also there. So temperature should be 77 degrees Celsius and at least contact time of 5 minutes. Okay, so iodophore is self-indicating. Chlorine is the cheapest, most common. Quaternary ammonium compounds, cationic in nature, incompatible with anionic detergents, and peracetic acid does not leave any residue. Okay, so summarizing everything what we studied today. So firstly, we studied about the cleaning and sanitization. Cleaning means removal of soil and sanitization means reducing the microbial load. Cleaning should precede the sanitization. First we will go for cleaning, we will remove the extraneous material, then we will go for sanitization or reduction of the microbial load of the compound, microbial load of the equipments. Okay, then next we studied about the milk stones, which are acid soluble. We studied that milk stones are made up of calcium phosphate, calcium carbonates, and also denatured protein and denatured fat. So how we will remove them? By using acid soluble compounds, acid soluble detergents. Okay, so we need to use nitric acid, phosphoric acid, and not alkalis. Then we studied about the 4 types of detergents and how they will use. They are alkaline in nature, acidic in nature, neutral, and enzyme. And alkaline ones are mostly used in dairy equipment. Then chlorine is the most used sanitizer because it is broad spectrum, it is fast acting, it is cheap, economical, effective, cost effective, easily available, but it leads to it gets inactivated against organics and high temperature. So what we need to go for, we need to first remove the organic substances, then we will go for using of sanitizer or the chlorine. Then next, iodophores, these are also self-indicating in nature. They will convert into pale color once they are exhausted, and ppm 12.5 to 25 is used. Again, we need to avoid more than 50 degrees Celsius temperature, and tea dips can also be used with iodophores. Then we also studied about the cleaning in place, how temperature, chemical concentration, mechanical action, and time. All these 4 are the important key parameters to ensure that the cleaning in place is being conducted and whole of the equipments are being conducted and cleaned using these cleaning in place method without dismantling of the equipments. So today we studied about a very small topic, cleaning and sanitization, which is, even though it is a very small topic, but it is important for exam, and so we studied this. So next topic we will study about the special milks. We'll study about it in tomorrow's class. If you have any doubts, you can ask me anytime. So I'm closing this session now.